In the previous video, we have discussed that aberrant upregulation of this MLKL gene will cause metastasis of the pancreatic tumor cells. In this video, we will discuss the results where they have discussed that upregulations of this or aberrant elevation of this MLKL gene will cause the necroptosis, and it is not dependent on the pathways that involve RIPK3. So in the panel A, what they did is they uh, generate the organoid, or they did the experiment on the organoid with upregulations or overexpression of this MLKL gene and with the inhibitor of this MLKL gene. And what they have find out is that when they use this inhibitor, the tumor's cells goes down. That can be seen here, and also that can be seen that can be seen in this second row, and it can be compared it with the first row where organoid is continue to grow, whereas they do not see a significant decrease when they use this RIPK3 inhibitors. That can be seen here. Comparatively, this GW is the inhibitors that they have used. Its organoid size is continue to increase with time. And when they use this GSK872 and necrosis 1, they do not see too much difference when they compare with both of them. So only when they use only for this MLKL gene inhibitors, they can see the reduction in the tumor volume. And these things they have checked using Western blot also, where they have seen that when they use this when they in the case of this Western blot of the following samples that they have used in the panel A, what they have seen is that PRPK1, which is the indication for this necroptosis, is increased in the second row, where they have used the inhibitors and also for this third row and fourth row, it shows that it's the upregulations or phosphorylation RPK1 indicates the upregulations of this MLKL gene that can be seen here. However, for this necrosis one, they do not see the necroptosis is upregulated. So indicating that for this NEC1 pathway, or it is also related to RIPK1 and 3, where they do not see the necroptosis is upregulated, whereas for this second case and the third case, whereas for this GSK872 and GW is the inhibitors for the MLKL gene, they can see that there is a upregulation of this necroptosis pathway through this PRPK1 signaling signals in the Western blot. And also when they check the relative volumes, they can see that relative volumes in the case of the second row, that is PDO, and in the case of this third row, that is when they use the inhibitors for RIK, PK1 and 3, is low compared to the case of control, where is the first row and the last row, where the first row and the last row. And this one also shows the although in the figure it does not show the reduction in the volume, but when they do the quantitative analysis, they can see that its volume is decreased relative to the initial volume. That can be seen here, or we can say that increase in the relative to the initial volume is low compared to the case of the first and compared to the case of the second, where they have used first one is the does not have any overexpression of MLKL, and the last row has this MLKL gene inhibitors. So in this case, they see that increase in the volume is quite high, whereas for the other case, the increase in volume is not much. So I want to clarify this thing, that in the initially what I have said is might be confusing. So that's why I have clarified this thing again. And the reasons that they have find out in the cell cultures images is that when they use this MLKL gene, they can see that there is a rupture of the nucleus can be seen here, and both in the case of PANC1 and also in the case of ASPC cells. Whereas when there is no overexpression of MLKL, they do not see any ruptures of the cells, so indicating that cells are rupturing. Therefore, intercellular components might be moving out of the cells, and that's why they check for the PANC1 and ASPC1 different, since they see the since they saw the image or evidence of the cell structure in the both in the case of PANC1 and ASPC, they need to check the whether in the cellular matrix they can see some of the apoptosis related genes or not. And in this case they have find out that apoptosis related genes such as caspase-3, caspase-8, and necrosis related gene, that is PMLKL1, are found in this PANC1 cell and also in the ASPC cells, where MLKL1 genes are overexpressed. And these expressions, some of these expressions seems to be higher in the control case, that is where there is no MLKL gene overexpressed. For example, these phosphorylated MLKL signals are high where MLKL gene is overexpressed, and ASPC where MLKL gene is overexpressed compared to the control case, indicating that necroptosis is triggered in where MLKL genes are overexpressed. But the signals for the apoptosis does not seem to vary too much. And when they did the quantification analysis of some of the other intercellular components that are found in the extracellular matrix, they have seen that for the first they check the cell viability. So cell viability, when they use this NEC1 inhibitors, the cell viability increases because cell viability increases. However, when they use this when the overexpression of MLKL's genes are there, then they see that cell viability are reduced. However, when they use this inhibitors, when they use the inhibitors in that case, this is the inhibitors, they do not see any cell viability decrease in the cell viability. That is overexpression vectors with MLKL with inhibitors and overexpression vector without MLKL with inhibitors, do not see too much difference. However, when they use only the MLKL gene overexpression, they see the decrease in the cell viability. And same things that they have observed when they overexpress this NEC1 also, that can be seen here. However, when they use this NEC1 inhibitors, overexpression of MLKL and NEC1 inhibitors, they do not see a decrease in the or increase in the cell viability, indicating that this MLKL gene overexpression of MLKL gene is not being affected by RPK3 pathways, because GSK2 inhibit the RPK3 pathways. And similar results they have observed for ASPC also, where when the overexpression of MLKL leads to the reduction in the cell viability. However, when the inhibitors are used, they do not see any reductions. And when they use the inhibitors for the NEC1 and the overexpression of MLKL, the results, the cell viability does not increase, indicate that MLKL upregulations or MLKL genes does not depend on RPK3 pathways. And when they check the other intercellular components in the extracellular matrix, they can see that the increase in the ATPs increase in the ATPs when they use MLKL gene compared to the vector case. However, when they use these inhibitors, they do not see any increase in the ATP. And same things they are observed for ASPC, that is when they use MLKL gene, they see the increase. And however, when they don't use, when they use the inhibitors for MLKL, they do not see any increase. Indicates that necroptosis are low. And for this case of necroptosis 1, they see the increase in the ATP. However, when they use the overexpression of MLKL with inhibitors of necroptosis 1, they still see the increase, indicating that MLKL genes are not affected by RPK3 pathways. It is independent of RPK3 pathways, and the similar results are observed for this HMGB1, where in the case of MLKL inhibitors, they do not see any increase in the intercellular components in the extracellular matrix. However, when they use this overexpression of MLKL or the inhibitors for NEC1, an overexpression of MLKL, they can still see the increase in the intercellular components in the extracellular matrix, both for the case of PANC1 and ASPC1. And the quantifications and the Western blots of these figures, these quantifications are shown in the panel G, where the results can be seen that the phosphorylated MLKLs are upregulated when they use either NEC1, when they use this NEC1, and are decreased when they use the inhibitors for MLKL. That can be seen in this case, and similarly in this case the phosphorylated MLKL are low compared to the case where GSK872 is used. It is the inhibitors for NEC1. So when the NEC1 inhibitors are used, they can still see the necroptosis signals are high compared to the case where MLKL genes inhibitors are used. And so this is what the main things that are observed from this Western blot. And for this RIPK pathways, signals are different in the case of ASPC. We can see that RIPK3 pathways are upregulated, whereas in the case of PANC1 we do not see RIPK pathways are upregulated. In the panel G, finally what they did is they check the overexpression of MLKL gene with this necrosis pathways, GSK872 inhibitors for NEC1 and the GW inhibitors for MLKL. So when the inhibitors for MLKL are used, they see that the red signals or the necroptosis is reduced, whereas for the NEC1 and GSK872 they do not see any decrease in the signals. These results are same for the PANC1 and also for the ASPC, where we see the decrease when the MLKL genes are inhibitors are used. And the quantifications of these fluorescence images are shown in the right hand side, where we can see that when the MLKL inhibitors are used, the dead cells are almost negligible compared to the control. Dead cell difference is almost negligible compared to the control. However, where MLKL genes are upregulated, the dead cells is increased. And similarly, when MLKL genes are upregulated and NEC1 or RIPK pathways inhibitors are used, they can still see the dead cells, both in the case of ASPC and in the case of PANC1. So all this indicates that necroptosis are mediated by aberrant elevation of this MLKL gene, and it does not depend on RIPK3 pathways. And if you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.